All right, good afternoon, everybody. I'm going to show you some learning effects in extremely young brains in my presentation, some recent results from our group. Early development of human cognition is still a fairly evolving field in neuroscience. It's quite hard to investigate altogether, because you have to be very careful with the methods. They have to be completely non-invasive and safe, and also behavioral methods are very hard to apply. Head-turning responses have been used, and they have provided very interesting results and important information, but this is a fairly unreliable approach. One way to study young brains, infant brains, is to use event-related brain potentials, which is certainly a non-invasive and safe approach for this purpose. And you can get temporally accurate information, which means that you can observe the neural processes with high time accuracy. And that way you can also acquire information on stimulus-specific processing. With EEG, you can study newborns, and if you want to go beyond that, you can apply MEG, for instance, a flat bottom device or a device with which is designed for investigating fetuses. And I'm going to introduce to you now the mismatch negativity response, which was used in these studies. It was discovered by Näätänen and colleagues in 1978, and it reflects auditory and linguistic processes. There are partly different generators in the brain for speech-related MMN processes and for acoustic processing, so these processes can be disentangled. This response is an index of sound discrimination accuracy and learning-induced plasticity. And the important thing is that it's elicited even without the participant's attention. So in that way, it's a very feasible tool for studying infants. And indeed, it is also a very ontogenetically early response, so you can detect it from the infant brain. And this is now to illustrate, this is a schematic illustration of the mismatch negativity. Oh, good, I see, get this here. So when you present sounds to a participant, they elicit in adults a response like this, as shown with the dashed line here. So there is first the P1, then N1, and then P2. And if there is a change in the sound sequence which is perceivable for the participant, then an MMN response is elicited, which you see here. And it's usually studied from difference waves. You subtract these two responses from one another, and then you get this kind of a response. You need lots of replications and repetitions for acquiring this response, so you have to collect data over a long period of time to get a reliable response. And this response is usually when you use EEG and record potentials over the scalp. Then it's largest in adults over frontocentral areas, and it's generated in the temporal lobes, and additionally there are generators in the frontal lobes, in the parietal areas, and even in subcortical areas. I will first present results from an adult study to demonstrate how this response reflects speech, sound processing, or memory traces for speech, native language speech sounds, and foreign language learning. A study by Winkler et al., in which a stimuli Finnish vowel contrast e versus ä was used. And MMN was recorded while the participants were reading a book, not paying attention to the stimuli. And then there was a separate session in which discrimination of these sounds was tested. And participants were Finns, Hungarians who had no Finnish command, and Hungarians who were fluent in Finnish. And let's first see the results from the behavioral test. So this was an identification test. And as could be expected, Finns were identifying their native language vowels with high accuracy, whereas Hungarians who had no command of Finnish were performing at chance level. Hungarians who were fluent in Finnish, they had been staying in Finland for several years, so they really mastered the language. They were almost as good as the Finns. And reaction times showed a consistent pattern, so that Hungarians not knowing any Finnish had longest reaction times. And let's then see what the MMN response is telling us about these processes. So these responses were recorded while the participants were not really listening to the stimuli. So we are talking about automatic neural processes. And here, this response is from Finns, and they show a nice MMN response for the native language vowel changes, which is what could be expected. They had representations in their brains for these sounds. Hungarians have no MMN response whatsoever. So neurally they could not make a distinction between these sounds. And if we look at this orange curve here, the Hungarians who had acquired the Finnish command actually after childhood, they have a similarly nice response as the Finns. So this is a learning effect even after childhood. You have acquired a mastery of a language, and then your brain becomes sharp with these sounds and even automatically reacts to changes in these sounds. Okay, then a few words about the development of the MMN before we go to the proper results concerning learning in infancy. The MMN, or it's also called mismatch response, is generated in the brain since the fetal stage. However, it changes during the development, and probably the first mismatch responses in the fetal stage were reported by Huotilainen et al. In 2005, and on the same year, Draganova et al. Also carried out a study on fetal MMNs. And this is to show how the recording happens and what's happening in the brain of fetuses. So there's the mother and the fetus, and then you place a flat-bottom MEG device on the abdomen of the mother. And here is the auditory cortex, and you present sounds to the fetus, and every now and then there are changes. And for these changes, it's possible to acquire a neural response from the fetus. Mother is wearing earphones at the same time, so that mother's reactions are not affecting the fetal responses. And a few words about the development of MMN. So already a newborn or even a fetus has an MMN or mismatch response to various types of sound changes, both speech-related and non-related. And here are results from a study by Partanen et al. And here are responses from newborns for vowel identity changes and vowel duration changes. And these are two conditions which is not relevant here. But as you see, there are systematically now positively displaced responses in these newborns. And if we look at the development, then for instance, a very nice study by Trainer et al. Suggested that at the age of 3 to 4 months, the mismatch response starts to change its polarity and become more adult-like. And at 6 months of age, infants have, most of the infants have an MMN which is resembling the adult MMN. However, stimuli and filtering, experimental parameters may have also an influence on these components. And this is just this figure here by Niemitalo-Haapola et al. Demonstrates that at 2 years of age, children, can you see the responses? They are not very clear here. Children have an adult-like MMN response for various types of changes. Here there are a consonant change, a frequency change of a syllable, intensity change, a vowel change, and vowel duration change. And here, when you use large contrasts, then you get a prominent response which is having a fairly similar scalp topography as in adults. Okay, let's go to the learning studies. So when a newborn is coming to this world, he or she is not a tabula rasa, really, since in the womb, infants get lots of stimulation. They don't get visual stimulation, that's pretty much blocked, but they get tastes through the liquid of the womb, and it's relevant what mother is eating, so they have experiences on tastes. And they also get somatic sensations since they move and they touch the surroundings and also themselves. And they get lots of sounds from, first of all, mother's body and also from the surroundings. So when mother is speaking or somebody is talking around nearby the mother. So they have experience on stimulation. And we actually then asked in this study carried out by Partanen et al. That what's happening neurally and can fetuses learn language? And in this study, there were two groups of fetuses. One was hearing a CD in which a pseudo-word, a Finnish pseudo-word, tatata, was presented. And there were every now and then a vowel or frequency changes. And that happened 5 to 7, or this CD was played 5 to 7 times per week, starting on pregnancy week 29, and it was continued until birth and not after that. And the control group got no stimulation at all. And then mismatch responses, when these infants were born to this world, then mismatch responses were recorded so that there was a repetitive standard stimulus tatata, and then various types of deviants. Some of them were those which were played to the training group, and some were control stimuli. And this is to show one of our participants. And what was found in these babies when they were born were indeed mismatch positivities, consistently with previous studies reporting positive mismatch responses. And the next figure will show in more detail what our results were. I will now start here with these vowel identity and vowel duration changes. These here represent the strength of the mismatch response. So the larger the bar, the stronger the mismatch response, and the better the brain discriminates the sounds. This is the basic idea. And the black bar is representing the activity from the learning group, and the gray bar from the control group. And let's look at the vowel identity and vowel duration changes, activations which were elicited by these changes. Finnish language is very rich with vowels, and we also have, it's a quantity language, so there are vowel duration changes which are important. So what we actually expected was that we do get both from the training and the control group significant and reliable mismatch responses for these changes, since this is what the fetuses are hearing in the mother's womb. And this is exactly what we found for vowel identity changes in both groups. The mismatch response was reliable, it was there, and for vowel duration changes, it was significant in the training group and nearly significant in the control group. And this is now the important contrast here, this pitch change. This is something, this change is something that we don't usually hear in the Finnish language. So we have either a steady prosody or a descending one, so the frequency is going down in each word. And here there were higher frequency syllables in the middle of the word, and that sounds quite odd to a Finnish ear. The training group was hearing these stimuli. I've lost now the cursor, where is it? It doesn't matter. Yes, okay, there. So training group was hearing in the training material these types of changes, and indeed we found a strong MMN or mismatch response for these changes in this training group. It was significant, and the activation was significantly different from that of the control group. Surprisingly to us, also the intensity changes, which were not included in the training material, activated the brains of these infants who were in the learning group. It was interpreted so that there was some kind of generalization. Perhaps when these fetuses were hearing these ta-ta-ta sounds, they were paying attention to several features happening in them. I don't know, that might be the explanation. Nevertheless, when there was a control condition where harmonical tones were played to these two groups, there were no differences between the groups. So they had similar kinds of basic auditory skills. Okay, the next question is that since these fetuses seem to learn, can they actually preserve these memories for a long period of time? Do these learning effects last? And this was tested in the second experiment carried out by Partanen et al. Again. And here, not the MMNs, but basic responses for sounds were recorded. And in this study, there was a group of fetuses which was hearing Twinkle, Twinkle, Little Star 5 times per week, starting on pregnancy week 29, continued until birth. And again, there was a control group which received no stimulation, and the recording was carried out again after 4 months. Let's first look at the results right after birth. So there are neural responses for the Twinkle, Twinkle, Little Star, and as you see, there is a group difference. There is a stronger response in the learning group than in the control group. But even more surprisingly, here are the results after 4 months. What we see is that the response has changed, so there has happened maturation in both groups. Interestingly, there is still the difference between these two groups. So indeed, these infants who had been hearing Twinkle, Twinkle, Little Star in the womb, they still had memory representations for this melody. Okay. This was it. So I summarize. We can use ERPs and mismatch responses to illuminate early stages of infant development. And the environment has an influence on the infant even before birth. And we have to keep in mind that all sorts of sounds may affect then the infant brain at that stage. Even harmfully unstructured noise sounds might be influencing them. Fetuses learn about the sounds and speech of the environment, and amazingly, the memory traces of sounds learned before birth may last for at least several months, at least for 4 months, which was the follow-up period in our study. And I'm finishing by thanking the colleagues and main collaborators. So Dr. Eino Partanen was responsible for the infant studies, and involved were also professors Risto Näätänen, Istvan Winkler, Paavo Alku and Minna Huotilainen. And thank you for your attention.
Questions, please. Andre.
Very good talk. Thank you very much. Such exciting data.
Thanks.
It's a shame.